Hello everyone, Mr. Linder here. Let's talk about excitation contraction coupling in cardiac muscle. So to start out with, I want to discuss a couple concepts that relate to cardiac muscle cells. The first thing to understand about cardiac muscle cells is about 1% of cardiac muscle cells are actually part of what we call the conduction system of the heart. These are going to be your autorhythmic cells. And so autorhythmic cells are essentially what you might refer to as pacemakers. And so these 1% of cardiac muscle cells are actually found within specialized areas of the heart, and they actually send electrical signals throughout the heart without the influence of the central nervous system, and they control the other 99% of the cardiac muscle cells. Now, of course, that's not to say that the cardiac muscle can't be influenced by the nervous system. It actually turns out that the autonomic nervous system, your sympathetic and parasympathetic branches, can influence the rate of your heart. But predominantly, the rhythm of the heart is put in motion by these 1% of cardiac muscle cells that we refer to as the autorhythmic cells or pacemaker cells. You may be familiar with some of the terms. So there's an area of cardiac muscle cells, these 1% of cells, known as the SA node. Also part of the conduction system, we have the AV node. And so information is going to travel across the atria as that information travels towards the AV node. From the AV node, we have the AV bundle, and then that branches into the right and left bundle branches. And then we also have fibers that run up the walls of the myocardium, known as the Purkinje fibers. So again, we have the AV bundle. That's going to be this section right here. And then we have the bundle branches. And then lastly, we have those Purkinje fibers. And so these all make up the conduction system of the heart, and they make up about 1% of the cardiac muscle cells. And so these are sending the electrical signal across the heart, and that's what's controlling your heartbeat. The other 99% of cardiac muscle cells are the contractile cardiac muscle cells. And so those contractile cardiac muscle cells are the ones that are responsible for pumping blood. So essentially, you've got 1% of the cardiac muscle cells in your cardiac muscle tissue that are sending electrical signals. And then the other 99% of cardiac muscle cells are essentially contracting so that you can pump blood through the heart and then through the pulmonary circuit and the systemic circuit. When you look at cardiac muscle cells, you'll notice that they're actually very similar to skeletal muscle. So it has a striated appearance. That striated appearance comes from the fact that you have sarcomere structures. And sarcomeres contain predominantly actin and myosin, and the actin and myosin proteins are arranged in a specific way so that you have this striated pattern when you look at the cells, and they're also arranged in a specific way so that you can have muscle contraction. And you can watch other videos on sliding filament theory. And so that's why you see this sort of dark and light banding pattern that repeats itself over and over again. So you have dark, light, and that's that striated appearance. Cardiac muscle cells have a single nucleus. You'll also notice, though, that cardiac muscle cells have intercalated discs. Intercalated discs are made of gap junctions, and those gap junctions are allowing for communication between the cardiac muscle cells And so what you end up having is this idea of a functional syncytium. The cells are interconnected with one another, and so they can work and they are coordinated as a unit. You'll also notice in cardiac muscle cells that there are lots of mitochondria. So there's mitochondria scattered throughout this image, and those mitochondria obviously are for producing ATP, and that's to ensure that you can continually have muscle contraction in an aerobic way inside cardiac muscle cells. In fact, cardiac muscle cells consume a large amount of oxygen on a daily basis so that they can maintain aerobic metabolism. If we want to look, though, at the actual contraction of these cardiac muscle cells, we need to look at the idea of the excitation contraction coupling that takes place in these cardiac muscle cells. So what we're doing here is we're zooming in on the plasma membrane of the cardiac muscle cell. So this would be the plasma membrane of a cardiac muscle cell, and this would be the intracellular environment. So we have extracellular out here, and then we have intracellular here, and this is the plasma membrane. And so what's taking place as an electrical signal moves along the conduction system and actually activates these 99% of cardiac muscle cells that are going to pump blood for the heart. So we know that the signal originates from the SA node, and when we're talking about a signal, we're talking about an action potential signal. So action potentials represented by this red arrow here. This electrical signal is going to be traveling along the plasma membrane of the cardiac muscle cells, and it's going to go down into the cell in this invagination structure called a T-tubule. So T-tubules are an anatomical structure that you're going to see in skeletal muscle, and we see here in cardiac muscle, and they are invaginations into the cell. So we're following the plasma membrane, so we can bring that electrical signal deep into the cardiac muscle cell. So the action potential signal is traveling along the plasma membrane and down into the T-tubule structure. So we're moving that electrical signal along the plasma membrane. As the electrical signal moves into the T-tubule, we're going to activate calcium voltage-gated channels. And so right here we have a calcium voltage-gated channel. This is an L-type calcium channel. And that channel is going to open because we reach threshold with the action potential signal. And that's going to allow this extracellular calcium to start entering into the cardiac muscle cell. So we have extracellular calcium entering into the cardiac muscle cell. That calcium is going to interact with a second type of channel. These are ryanodine receptor calcium channels, RYR for short. And these calcium release channels are actually in the sarcoplasmic reticulum. So SR stands for sarcoplasmic reticulum. Write that out for you, sarcoplasmic reticulum. And really, sarcoplasmic reticulum is just talking about smooth ER. This is smooth endoplasmic reticulum, which are very good for storage. And so we have calcium storage within our cardiac muscle cells. And we are essentially using the calcium voltage-gated channels in the T-tubules to activate the RYR channels. And when those RYR channels are opened, then we can see calcium being released into the extracellular fluid in larger quantities. So that stored calcium in the sarcoplasmic reticulum is released and it begins to generate what is commonly referred to as a calcium spark. What's interesting about the calcium spark or sparks is that you can have summation. meaning that you can add together this calcium that is flowing into the intracellular fluid now. So we have extracellular calcium coming in, we have calcium being released from the sarcoplasmic reticulums, and we are adding together all of this calcium in the intracellular fluid to generate a calcium signal. So we need a strong enough calcium signal in order to activate cardiac muscle contraction. The calcium signal is essentially diffusing. So calcium is diffusing through the intracellular space. It's moving into the sarcomeres. So here we have the sarcomere structures. And again, the sarcomeres are predominantly made of actin and myosin. So the actin is in red, and then the myosin has these sort of globular head structures. And so the calcium signal, the calcium ions are moving to the sarcomeres, diffusing, and they're going to bind with a protein known as troponin. So troponin is actually found resting on a protein called tropomyosin, and both troponin and tropomyosin sit on the actin protein. And what this is doing is, when calcium binds troponin, is it's moving the tropomyosin protein so that myosin and actin can bind with each other. And when myosin can bind to actin, these globular head structures can go through what's called a power stroke. And the power stroke is basically the movement of that globular head, and that's going to cause the actin to slide over the myosin. And so when actin slides over the myosin, we get muscle contraction. And again, these are the same types of events that you would see taking place in skeletal muscle. So electrical signal triggering calcium voltage gated channels that are tied to triggering the ryanodine channels, release of calcium from the sarcoplasmic reticulums, a summation so that we get a calcium signal. Calcium binds troponin, moves tropomyosin, and allows for myosin and actin to bind each other. And when myosin power strokes, we get that sliding filament mechanism, which is the contraction that takes place in the muscle cells. And so what that is this is excitation contraction coupling. Coupling means to join something together. So essentially what we're doing is we're joining the electrical signals from the conduction system with actual muscle contraction so that we can pump blood with our heart. In order to essentially stop this process, so the heart has to go back into a resting state so that it can begin to fill back up with blood. In order to rest the heart musculature, we need to essentially put calcium away. So relaxation is going to take place as calcium unbinds troponin. So we can see here calcium is releasing, so it's unbinding troponin. When it unbinds troponin, the tropomyosin protein can go back to its original position, which is going to block the myosin from binding to the actin. Then what we want to do is we want to put that calcium away. So we're going to use calcium ATPases. So we're going to actually spend energy to pump calcium back into the sarcoplasmic reticulum. So we're storing calcium back in the sarcoplasmic reticulums. We're also going to remove calcium from the intracellular fluid. So here we have calcium in the intracellular fluid, and we're going to use a calcium and sodium antiporter. So calcium is going out as sodium comes in, so that's an antiport process. And so that's going to move calcium back into the extracellular fluid. And then we also utilize a sodium-potassium pump, so spending more energy, to put sodium back into the extracellular space to keep this antiporter functioning. So we're restoring calcium and we're removing calcium from the intracellular environment so that cardiac muscle can go back to its resting state. The last thing I want to mention about this process is that cardiac muscle cells can contract in a graded fashion. Basically, what that means is that there can be varying amounts of force that are generated by cardiac muscle. So cardiac muscle can contract lightly, or cardiac muscle can contract heavy, depending upon the available amount of calcium. So in essence, if there are low amounts of intracellular calcium, so if you have low intracellular calcium, then you're going to see weaker contractions of the heart. If you have high levels of intracellular calcium, then you're going to see greater forces of contraction of the heart. So that's a graded type of contraction where you can have minimal amounts of contraction or stronger amounts of contraction based on the available calcium. The other way you can change the contractive force of the heart is by changing the overall stretch of the heart. And that's something that'll be discussed in another video, looking at something called the Frank-Starling law. So the Frank-Starling law of the heart is actually another way to change the force of contraction of the heart. So we can change the force of contraction based on available calcium, and we can also change the force of contraction of the heart based on how much blood enters the heart during a cardiac cycle. So that's a little bit of information on excitation contraction coupling and the types of cardiac muscle cells that you see in the heart, both conduction cells and the cells that pump blood. I hope that helps. Take care.